Isn't that amazing? All right. The last concept that we are looking at is attempting to sequence an unknown piece of DNA. Okay? And so there are lots of methods to do that. This is probably one of the most traditional methods. It's called the Sanger dideoxy method. Okay? Why in the world is it called dideoxy? Yes, we use a nucleotide that has two. Both the 2' and the 3' are hydrogens. And you need the 3' to continue the sequence and connect the DNA together. And so what we're going to do is we're going to use DNA polymerase. We're going to make copies of your unknown. And most of the time you're going to use a perfectly fine trinucleotide. And you're going to add, and you're going to grow, and you're going to grow, but every so often you're going to add in a dideoxy. When you add in a dideoxy, the synthesis stops. Okay, it's shortened. All right? And we do this lots and lots of times. And if your ratios of the dideoxy are correct, then you should get a very long sequence, up to thousands of base pairs long, where there is a stop at each base pair because you've inserted a dideoxy randomly. Okay? But then I've got a whole mixture of all of these DNA sequences, all different sizes. So I put them on a, in this case, an acrylamide gel. I run them such that the resolution is I can separate the pieces of DNA that are only one base pair different. So base pair that has 9 and base pair of 10 will separate. And then I can read the sequence of the DNA. Now, there are two different techniques. We can do it in 4 test tubes, or we can do it in 1 test tube with fluorescence. Most of the time, people will do it in 1 test tube with fluorescent markers where the dideoxys are fluorescent, and there's an instrument, kind of costly, but that was able to detect it. I'll show you that. But if you don't have the very, very costly instrument, you can do it in a 4 test tube method, and so I'll show both of them. Now, the animations will be on the Moodle site. In this case, sometimes it gets confused, and it doesn't know what the peak is, and it'll put an in, and so sometimes you get a non-red base pair in that particular sequence. Okay. And so here's a picture of what that would look like. It's 1 lane. They aren't colored as they go through. They actually look clear only when the detector and the fluorescence excitation happens will we see, will we be able to detect the different fluorescent colors. And what you do is you get a readout with the base pairing being this one would be 5', this is 3'. Okay, this is the sequence of the new DNA, not our original, so you have to translate that to get the original unknown DNA. Okay, different colors. Okay, we got that. Already said that. Actually, what that represents, let's go back 1 second. This right here, it's an overlay of 4 different readings. What it really is, it overlays, and for each position, it measures how much blue, how much green, how much yellow, how much red is there. And what it does is it represents it on top, so you see 1 peak at each position. And so that's really what's going on when you read this. Synthesized, the original, and you got to watch out. On the exam, the original could be written 5', 3', but most often the letter A, your answer, will be written 5' because that's traditional. We write 5' at the front. So this strand and this strand are exactly the same. It's just almost all the time we write 5' first. So don't let that confuse you either. But that could be a little twist in the thing when you're trying to translate this and you're on the exam and you're, So watch out for that. All right. Another technique that we use to take a piece of DNA and extract it is the polymerase chain reaction, or PCR. It's actually kind of interesting. All right, if you're trying to purify something, right, that means you usually separate it out and you purify. PCR can actually purify something without taking anything out of the test tube. Okay, and so if by chance you have a whole genome, but you only want one gene, okay, well, you've got the whole genome and I have one gene. What I'm going to do is I'm going to create a tag that I know the sequence on this side of the gene, and I'm going to buy a tag on the other side of the gene, and using just these primers, your original piece of DNA, and a heating and cooling block, I can get a pure sample without separating anything. Okay, I'll show you how that is done. So if you took your pipette and put it in that sample, and you pipetted out something, essentially, statistically, you'll only get the target. You won't get any of the others. So it becomes essentially pure, just by statistics. Now, this was awarded the Nobel Prize, this technique, because it was so powerful, and it was all because of surfing. The person who developed this was a grad student, and his professor wanted a large portion of DNA of a particular type. And up to this point, you had to grow cells, you had to cleave it, you had to purify it and isolate it little by little to get enough. This guy wanted to go surfing. And so he came up with this on a napkin in a bar, and originally, every time that you heated to melt the DNA, you would actually heat the protein, the polymerase, and he'd kill it. So he'd have to squirt in more polymerase after that round to keep it going. Now that was a whole lot simpler than growing up cells and isolating DNA. Now they talked about using a particular polymerase called Taq polymerase. Taq polymerase is just an isolated polymerase from one of those organisms that live in waters that are at near boiling temperature, in which its function is still fine at 100 degrees, and so it was able to survive that heating and cooling cycle without having to re-add that protein in. And so Taq polymerase is really a commercial version that has been patented. And so this technique cost us money because someone was making a lot of money off of using that one particular enzyme. All right, last thing, gene expression analysis. Basically, a couple techniques to do this. A lot of times when you're trying to look at treatment options and you want to look at what's happening in the cell, all right, this is a more complex idea. And so let's say you have a cancer cell, and you want to know what happens if you treat it with a drug. Okay? What you really want to know is there any difference between non-treated and treated? Is something being affected? All right. What you really want to do in terms of cancer cells, you want to downregulate something because the growth. Well, to do that, we have a couple techniques where what we do is we treat the two cells. Okay, one a control non-treated and an experimental treated. And then what we do is we isolate the messenger RNA. Okay? Now, the messenger RNA is going to be an analysis at that moment in time what's being signaled for the protein to do, or for the cell to do. Okay? And so, if I can isolate those messenger RNAs, I'm going to try to analyze them and see what is the difference between the two. Okay? Well, and we're going to look at two different ways that we'll do that analysis. This particular animation is not on Moodle. It's actually in your textbook, not textbook, but the online homework site. So, just ways to do analysis. Again, many of these types of analyses are now in kits. All the enzymes are in there, and it's just got solution A, which is yellow, solution B, which is green, and it tells you how many drops to do, and it's all very optimized and automated, but the types of information that you can get is very, very powerful. The last slides are the non-animated, the versions of these so that you can see in picture form. All right, and that is the end of this particular chapter.